Hello guys, welcome to another video. In this course we will learn about plant breeding and the contents are here: history, role, challenges and objectives of plant breeding, scope and modern tools of plant breeding, hybrid, male sterility, mutation, plant introduction, pollination, centers of origin, gene pool, inbreeding and inbreeding depression, natural and artificial changes of traits, self-incompatibility, germplasm, activities in germplasm, apomixis, genetic erosion, heritability, heterosis. All of these contents will included in this course. So let's get started. What is plant breeding? Plant breeding is based on principles of genetic and cytogenetic. Plant breeding is the science of changing the traits of plants in order to produce desired characteristics. It aims at improving the genetic makeup of the crop plants. Improved varieties are developed through plant breeding. Its objectives are to improve yield, quality, disease resistance, drought and frost tolerance, and important characteristics of the crops. So this is the definition of plant breeding. Now I am going to tell about the historical background of plant breeding. We know that the history is very huge, so we need to just read the history. This is not our main concern. We can see here different types of scientists work in plant breeding, such as Camerius, Colruyter, Saal investigate effects of inbreeding and heterosis, male stereotype, Colruyter, Condell, Vavilov proposed 8 centers of origin. Then historical background is divided into different type of era, such as Premandelian Era. Here we can see 1665 to 1766. This duration is known as Premandelian Era, and here we can see that Robert Hooke, who is the father of cell biology, and different types of scientists work here during 1665 to 1766. We need to just study. This is not our main concern. Then after Premandelian Era comes Mendelian Era. Here we can see 1900 to 1919, and this duration is known as Mendelian Era. Mendelian Era, we can see here different types of scientists work here. Then Davenport proposed dominance hypothesis of heterosis. Then Saal first used the term hypothesis for hybrid vigor. These scientists are work here. We need to read the basic things here. So this is not our main concern. Then after Mendelian Era comes PostMendelian Era. Here we can see 1923 to 1945. This duration is known as PostMendelian Era. So, here we can see that different types of scientists work here. Then they investigate and they research very hardly, such as Sachs, Muller, then Vavilov, Rhodis, Golden. Different types of scientists work here, and they invent different types of things about plant breeding and genetics, such as mutation, male sterility, dominance hypothesis, heterosis. All the things comes from them. After the post-Mendelian Era comes modern era. Here we can see in that 1953 to 1983, this duration is known as modern era. Here we can see different type of scientists work here, such as Watson, Flor, Borlaug, Grafas, Donald, Patel, Borlaug, Haley. So we need to study this work of this scientist. So that's for today. We'll learn more about the plant breeding in the next video. So thanks for watching.